Good evening, students. Good evening. How are you all? Good evening. Good evening, students. Whether I'm audible? Yes. Good evening. Yes. 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 I hope up to this, no doubts are there. Okay. Today we are going to see fourth topic of our today's revision class. Okay. See, today we are going to see most important topics. Okay. So four most important topics I have bring today. Okay. First and foremost important thing is phytohormones. Okay. From phytohormones, if we analyze the previous year question paper, you will get minimum 2 to 3 questions. Okay. Minimum 2 to 3 questions you will get. And respiration, sure you will get 1 to 2. Okay. And in stress physiology, again I am saying some of the people will not read the stress physiology, but sure every year they are asking 1 to 2 question from stress physiology. And another most important thing, photomorphogenesis. That means phytochromes, cryptochromes. Okay, from there you will get minimum 1 question. So out of 10 questions, out of 100, 10 questions we will get from this lecture. Okay, so listen properly. Okay, I will give some of the hints also how to remember the phytohormones. Okay, what are the major questions they will ask. Okay, these all things we are going to discuss in today's lecture. Okay, this is just a revision class. Okay, I will just let you know what are the important points, what are the things you have to remember for exam point of view. Okay, first we are going to start with phytohormones. Okay, phytohormones. Before starting the lecture, I will tell what are the most important things you have to learn in the topic phytohormones. Okay, there are different classes of phytohormones are there. What are the things you have to remember means, first thing, what is the nature of the hormone, whether it is a terpenoid compound or what are, so nature of the component you have to know. Okay, second most important thing, biosynthesis. What is the biosynthetic precursor and what are the organelles involved in biosynthesis. And third most important thing and most important thing is you have to know the physiological function of each and every plant hormone. Okay, what is the major physiological function of each and every plant hormone. And fourth important thing, you have to know about the signaling. Okay, if this hormone is there, how the physiological functioning of the plant will be modulated, what are the major receptor and how the signal will be transferred in the plant. These are the major things you have to focus. Apart from that, you also have to read about the bioassay. Okay, if an hormone is there, what are the different bioassays to determine that one. Okay, so these are the key points you have to remember for this phytohormones. Okay, first we will see what is meant by phytohormones. Okay, the basic definition of phytohormones are what? They are naturally occurring organic compounds. Okay, so phytohormones are naturally occurring organic compounds that regulate the plant physiological functions. Okay, that will regulates the plant physiological function at site distant from their production. That means it can mobile from one place to other place for doing its physiological function. This is the general definition of phytohormones. Okay, this phytohormones we can also call as plant growth regulator. This is another important point. This we can also call as a class of plant growth regulator. Okay, so plant growth regulator is a broad term. It will consist of both organic and inorganic molecules. Organic or natural or synthetic molecule will be there that will regulate the physiological process. Okay. Then another most important thing, the term phytohormone was given by Thiemann. This is JRF question. Okay. JRF 2019 they asked. Okay. So the term phytohormone was given by Thiemann. Okay. And the another most important thing, auxin is the first plant hormone or first phytohormone discovered. This is also another important point. Okay, there are two major categories of phytohormones are there. Okay, one we can say as growth promoters and second is known as growth inhibitors. The major example of growth promoters are auxin, gibberellin and cytokinin. Okay. Growth inhibitors are what? Ethylene and abscisic acid. I hope this is clear for everyone. Okay. Now we are going to see about the what is the basic chemistry, what is the major physiological function of each and every plant hormone. First, we are going to see the 5 major plant hormones: auxin, gibberellin, cytokinin, ethylene and abscisic acid. This 5 is very important. From these 5 hormones, you will get one question. Okay, we are going to cover this 5 hormones in within 30 minutes. Okay, just it's a marathon revision class. Okay, first synthesis or first discovered plant hormone is what? Auxin. Okay, we are going to see about the discovery and chemical nature of this hormone. Okay, discovery and chemical nature of this hormone. Okay, first we will see the scientist contribution. Okay, Charles Darwin and Francis Darwin, what they have done means they have studied the phototropism response using canary grass. Okay, using canary grass, they have studied about the phototropism response. Okay, now you have to know about what is meant by phototropism. Phototropism means what? Phototropism means when we give a unilateral light, the bending of the plant will be there towards the light. Okay, bending of the plant tips will be there towards the light. This is known as phototropism. Okay, so auxin is primarily involved in phototropism. This is another important point you have to remember. Okay, from his observation, what he had concluded, the colloidal tips will bend towards unilateral light. Okay, if we cap the tip with foil, or if we cap the tip with the foil, the stimulus will be prevented. Okay, so the tip will act as a stimulus for that phototropism. Okay, phototropism. This all things was published in the Power of Movements in Plant. This is another important point. Okay, this is the classical publication from Charles Darwin and Francis Darwin regarding the hormone auxin. Okay, then most important discovery was Boyson and Jensen. What he did mean, he has inserted a gelatin block. For example, this is coleoptile tip. He has inserted a gelatin block. Okay, this gelatin block will allows the transfer of phytohormones from this region to this region. Okay, so he has inserted a gelatin block between the decapitated tip and the stump. Okay, as a result, what he found, the coleoptile was still it is bending towards the light. So proving that stimulus is what? It is mobile in nature, that is diffusible in nature. This is the key conclusion was derived from Boyson and Jensen experiment. Okay. Another most important thing is Paul. What he did means he has placed the decapitated tip. This is a decapitated tip. He has placed asymmetrically. Okay. He has placed asymmetrically on the stump in total darkness. Okay. He has placed asymmetrically. What is the conclusion he have seen means that the tip of these things will secrete a growth promoting substance. Okay, he has concluded in this tip the growth promoting substance are producing, that is responsible for this asymmetric distribution of this auxin. Like that he has concluded. Okay, this is another important and most important and repeated slide. Repeated question they are asking from this slide only. Okay, the term auxin was given by F.W. Wendt. He is the person who was first successfully isolated the auxin hormone. Okay, first successfully isolated the auxin hormone. Okay, the major bioassay for auxin synthesis is what? Avena curvature test. Avena curvature is the basic test for determining the auxin content. Okay, these two are very important point. Okay, apart from that, there are minor hormones are there, like minor classifications of auxins are there. You just go through this one. This is not that much important. Just you go through. Okay, auxin A, auxin B, and heteroauxin will be there. And from where they have isolated and who is the scientist contributed. This is another important thing. This is the different experiment what they have done. You see, Darwin firstly he has concluded about the phototropic responses. Second most important thing is Boyson Jensen. He has placed the agar. Then he has concluded the stimulus is mobile in nature, and Paul he has concluded the tip is secreting some of the chemical substance, ठीक है, organic chemical substances synthesizing, like that he has proven. ठीक है, these are the some of the contribution. From this scientist contribution we can expect some of the question if they want to set the question paper difficult. ठीक है, now we are going to see the classification of auxins. ठीक है, auxin we can broadly classify into natural or synthetic one. You have to just remember what are the example of natural auxins, what are the example of synthetic auxin. Okay, so natural auxin example is what? Indole 3 acetic acid, indole 3 butyric acid, 4-chloroindole acetic acid and phenyl acetic acid. These are the example of natural auxin. Then what are the example of synthetic auxin? Tiba. Okay, Tiba is an example of synthetic auxin. Apart from that, NaO will be there. Okay, then 2,4-D will be there. These are the example of synthetic auxins. Okay, so the synthetic auxin 2,4-D can be used as an VD site. Okay, we can use that as an VD site. That is also another important point you have to remember. Okay, now we are just going to see what are the major physiological efforts of auxin. If auxin is there, means what are the major physiological function it will do. The first and most important thing is what? Cell elongation. Cell elongation will be done. This is the first and most important function of auxin. How? It will activate the H plus mediated acidification in the cell wall. Because of that, the cell wall will become loosened and it become elongated. Clear? So H plus mediated acidification was done by in auxin hormones. Okay, H plus mediated acidification will be done. Okay, so first major physiological function is cell elongation, and second most important thing is apical dominance. This is another important and repeated question. Auxin is the hormone primarily involved for apical dominance. Okay, so apical dominance means what? The presence of an intact apical bud. For example, here an apical bud is there that will suppress the growth of the lateral buds. Okay, so that will suppress the growth of the lateral bud. This is the simple definition for apical dominance. Apical dominance is primarily due to presence of auxin at the tips. Okay, then most important thing, another thing, root initiation. We know auxin is primarily involved for root initiation. At high concentration, it will inhibit the primary root growth, and low concentration it will promote the lateral root growth. Okay, this is another important point you have to remember. Another thing is callus formation. This we already seen in our first lecture itself. So for callus formation, we need equal concentration of auxin and cytokinin. For promoting the root, we need high concentration of auxin, and for shoot, we need high concentration of cytokinin. This we have already discussed. Okay, and the most important thing, abscission will be prevented, parthenocarpic fruits can be produced, and auxin can also increase the rate of respiration. Okay, then if you see the distribution of auxin in plant, the tip of the plant, for example, root tip and shoot tip will have high concentration of auxin. Okay, root tip and shoot tips will have high concentration of auxin. And another most important thing what you have to remember is the only hormone having polar transport is auxin. Okay, auxin is the only hormone that is having a polar transport. This is also another important point you have to remember. So polar transport will be present only in auxin. Okay, this also you have to remember. Now we are going to see the biosynthesis of auxin. Okay, from here they have asked a question from JRF 2023. In JRF 2023 they have asked the question. What question they have asked means they have given different pathways of auxin biosynthesis, and they asked in which crop which pathway will be there. Okay, if you see broadly the auxin biosynthesis is classified into two types. Okay, one is tryptophan dependent and tryptophan independent pathway. Okay, so the primary precursor for auxin synthesis is what? Tryptophan amino acid. Okay, tryptophan amino acid. Then another pathway is there, that is tryptophan independent pathway. This tryptophan dependent pathway was further classified into four types. This is the important point you have to remember. So total 4 classification are there. Tryptophan dependent pathway. First is tryptamine pathway. Then second is indole pyruvic acid pathway. These two are major pathways. These two are the major pathways involved primarily in auxin biosynthesis. Apart from these, two minor pathways are there. One is indole acetonitrile pathway. Indole acetonitrile pathway. This is primarily present in, this is the question they asked in 2023. Okay. This pathway will be predominant or primarily present in Brassicaceae family, Musaceae family. Okay. Musaceae family and Poaceae family crops will have this indole acetonitrile pathway of auxin biosynthesis. And the most important thing is IAM pathway. Then fourth pathway is IAM. This IAM pathway will be present in pathogenic bacteria, particularly if you see Agrobacterium tumefaciens and Pseudomonas savastani. Okay, these two will have this IAM pathway. So IAM is a bacterial pathway. We know crown gall disease is causing means due to presence of excess production of auxin and cytokinin in root cells. Okay, so these are the example of different pathway. I have just simplified here. You have to remember the key points. Okay, total 4 pathways are there: tryptamine, indole pyruvic acid, indole acetonitrile and indole acetamide pathway. And what are the major examples? Okay, what are the major example? Now we are going to see about the signaling aspect. Okay, this we already discussed. Polar transport will be there present only in auxin. Okay, then if excess of auxin is there means that has to be stored or it has to be degraded. Okay, this is another important point. There will be one enzyme IAA oxidase or peroxidase. This will convert or this will degrade the auxin to 3-methylene oxindole. This is another important point you have to remember. Okay. Now we are going to see about the signaling. Okay. Biosis, this we have already discussed. Okay. Signaling. We are going to see in presence of auxin and in absence of auxin. Okay. If auxin is there, what it will happen? If auxin is there, you have to remember only exam point of view. If auxin is there, the inhibitor OXIAA will be degraded by 26S mediated proteasome. So the auxin responsive factor will come and bind. Auxin responsive genes will be active. I hope this is clear. If IAA, if indole acetic acid is there, this will lead to degradation of OXIAA. This is the inhibitor molecule. If this inhibitor molecule is degraded, auxin responsive factor will bind here. Because of the transcription will be happen. If auxin is not there, auxin responsive factor binding will be inhibited. I hope this is clear. With this, we are completing the hormone auxin. Any doubt is there in auxin? Any doubt? Kindly let me know any doubt is there. No doubt, yes. Everything is clear. Yes, everything is clear. Okay. Now we are going to see gibberellins. Okay. Now we are going to see gibberellins. Okay. Gibberellin is second major class of phytohormone. Important things only we are going to see. Okay. So the gibberellin was first identified from where? Rice seedling. Okay. So Buchan disease. Buchan disease, this we can also called as foolish seedling disease. From that seedling, they have isolated the hormone gibberellin. Okay. Then another most important thing, gibberellin is a tetracyclic diterpene, that is, it is a 20 carbon compound. This is another important thing. They have asked in maths one time, okay, what are the different hormones and nature of the hormone, whether it is a diterpene, tetraterpene, sesquiterpene, like that they ask. So gibberellin is a diterpene molecule, that is 20 carbon containing molecule. Okay. And the key discovery, from the key discovery, what you have to remember means, Ebuta is the person who has isolated the active substance, that is gibberellin, and he is the person named the hormone as gibberellin. I hope this is clear. This they asked one time. Okay. Ebuta is the person who is isolated the active substance and named it as gibberellin. Okay. If you see the classification of gibberellin based on the carbon atom, they have broadly classified into C20 gibberellic acid and C19 gibberellic acid. This is just for your understanding. Okay. So the major example of C20 gibberellic acid is GA12 and GA53, GA12 and GA53. Okay. Then the second most important thing, C19 pathway examples are GA1, GA3 and GA20. Okay. GA1, GA3 and GA20. This is another important thing. Why I am writing here means this GA20, gibberellic acid 20 is there, na? This is the target. They have mutated to produce draft rice. Okay, draft rice. Okay, so if this gibberellic acid biosynthesis will be inhibited, if the gibberellic acid biosynthesis will be inhibited, internode elongation will be reduced, so the plant will become short. I hope this is clear. So in green revolution, they have done this one. Okay, GA20 oxidase enzyme they have targeted. Okay, I am writing here just for your knowledge. Okay, GA20 oxidase, GA20 oxidase. Okay, clear. This is very important point. Okay, next we are going to see the major physiological effect. Major physiological effect we are going to see. Okay, so if you see the major physiological effect, seed germination and breaking of dormancy. This is the primary role of gibberellin. Yes, seed germination will be there due to alpha amylase activity will be increased by the gibberellic acid. So primary role is seed germination and breaking the dormancy. And secondary role is what? Stem elongation and genetic dwarfism. I already told. If the GA20 oxidase was mutated, that will produce draft plants. Clear? That will produce draft plants. Okay. Then stem elongation. So that means internode elongation also will be there. In sugarcane internode elongation, we will spray gibberellic acid. Okay. This is the another important thing. Bolting and flowering. This is specific symptom or specific physiological function of gibberellic acid. Okay. Bolting and flowering will be done by gibberellic acid. Okay. Bolting and flowering by gibberellic acid. Okay. This definition, everything we have discussed in our class. What are the key important points? That is only I am going to say here. Okay. So parthenocarpic fruit we can be produced by application of gibberellic acid. Okay. Seedless grapes we can develop. Okay. Seedless tomato. These things will be developed by GA application. This we already discussed. Alpha amylase activity will be increased. Because of that, yearly seed germination will be there. Okay. These are the major physiological functions. Okay. Major physiological function of gibberellic acid. Now we are going to see the chemical forms of occurrence. Whether it will be present in free form or whether it will be present in bound form. Okay. If an excess of hormone is synthesized, that can be converted into bound form, then it will be stored in vacuole. Okay, clear. So whenever the hormone need is there, the bound form will be converted into free form, that will be utilized in physiological function. These are the different forms. Just this is for your understanding only. Okay. Now we are going to see the biosynthesis. See, in biosynthesis, what are the important point I am going to say? The precursor for gibberellic acid biosynthesis is acetate, or else we can also say as IPP. Okay, 3 molecular first chain will be binded to form IPP. So acetate or IPP is the precursor. And the second most important question what they will ask from gibberellic acid biosynthesis is, what are the organelles involved in gibberellic acid biosynthesis? The first organelle involved is plastid. Okay. Second, endoplasmic reticulum, and third is cytosol. So 3 organelles will be involved in biosynthesis of gibberellic acid. We are not going to see the each and every step, just outline I will give. Okay. So IPP will be formed in plastid. Okay. This IPP will be converted into geranyl pyrophosphate within the plastid. This is a 20 carbon compound. This geranyl pyrophosphate will go to endoplasmic reticulum. There it will be converted into entcourin. This is just for your understanding. Entcourin will form. This entcourin will be converted into gibberellic acid in the cytoplasm. I hope this is clear for everyone. The another most important thing what they will ask in exam means, what are the stage specific inhibitors of GA biosynthesis? Okay, if you see the stage 1 of gibberellic acid biosynthesis will be inhibited by Paspan AMO1618 and cycosol. This is repeated objective question. Okay, this is repeated objective question. And the stage 2 will be inhibited by paclobutrazol. Stage 2 will be inhibited by paclobutrazole, and stage 3 will be inhibited by cyclohexanitriones. These are the key points you have to remember for exam point of view. Okay. Now we are going to see the bioassay. In bioassay, the most important thing is draft contest and barley endosperm test. These are the major example for bioassay of gibberellic acid. Now we are going to see the last topic, that is what? Signaling. Okay. If gibberellic acid is there, so gibberellic acid receptor is what? GID1 is the receptor for gibberellic acid. Okay. If gibberellic acid is there, this will degrade the DELA inhibitors. Okay. DELA is a protein. This is a amino acid sequence. DELA proteins will be there. This DELA proteins will be degraded by proteolysis. Because of this, MYB transcription factor will go and bind gibberellic acid responsive genes will be activated. If gibberellic acid is not there, DELA repressor will be there, that will go and inhibit the GA responsive genes. I hope gibberellic acid is clear for everyone. With this, we are completing gibberellic acid. Any doubts, students? Kindly let me know any doubts. Any doubts? Just let me know any doubts. Safura, Svetaraj, Rama, Viral. Okay. What are the specific example? What are the specific example for pathway of auxin? I already told two major pathway. IPA and tryptamine pathway will be universal pathway that will be present in like tomato, whatever the crops existing, except this Brassicaceae, Musaceae and Poaceae family that will come under class 1 and class 2. That is IPA and tryptamine pathway. Okay. In class 3 typical example we can say banana, rice. These are the typical example for indole acetone nitrile pathway. Okay. I already told. Then fourth pathway is what? Bacterial pathway. TAM pathway is bacterial pathway that will be specifically present in Agrobacterium tumefaciens and Pseudomonas species. I hope this is clear. Ramer viral. This is clear. I hope this is clear. Okay. Now we are going to see about the third hormone, cytokinin. Okay. This is just a brief line how to read. Okay. If an hormone is there, it can be synthesized. If excess is there, means that can be catabolized or conjugated. Then transport thing will be there. Then that hormone will be perceived for signaling. The responsive genes will be activated, like that I have given. If the responsive genes are activated, major physiological function will be done, like that. This is just for your understanding. Okay, cytokinin. This is JRF 2023 and 2021 question. Okay. Okay. So the discovery of cytokinin. This is very important point. Discovery of cytokinin. Cytokinin was discovered through effort to identify the component that increase the growth of plant cell in culture. Okay. Which is the plant hormone that was discovered through the effect of identify the component increasing the growth of plant cell culture is cytokinin. Then second most important thing, the cytokinin or kinetin is a breakdown product of DNA. Okay. This is another important thing. Okay. It is a 6-furfuryl aminopurine compound. Okay. It is a breakdown product. It is a breakdown product of DNA molecule. Okay. Breakdown product of DNA molecule. Okay. Skoog is the scientist who has first identified this kinetin. So this kinetin is a what? It's a synthetic molecule. Okay. Synthetic cytokinin molecule. This we already discussed, the concentration of auxin and cytokinin required for callus formation, root and shoot formation. Why I am again saying the same thing repeatedly means these are the very important points you have to remember. Okay. If equal concentration of auxin and cytokinin is there means callus tissue formation will be there. If high auxin is there means root. If high cytokinin is there means shoots will be formed. Okay. Now we are going to see the example of natural cytokinin and synthetic cytokinin. The example of natural cytokinin is what? Transzeatin. This is very important. Transzeatin, isopentenyl adenine and dihydrozeatin. These are the example of what? These are the example of most active or natural abundant cytokinins. Okay. This synthetic cytokinin, this is JRF 2021 question. Okay. The example of synthetic cytokinins are what? Thiodiazerone and benzyl adenine. These are the example you have to remember. Okay. And the most important thing, now we are going to see about the biosynthesis. In biosynthesis, I will tell you what are the key points you have to remember. The precursor for cytokine biosynthesis is ATP or ADP molecule. I already told. Okay, DNA molecules will be there. So ATP or ADP along with dimethyl allyl pyrophosphate, or else we can say IPP. Okay, these are the major precursors for biosynthesis of cytokinin. The most biologically active form of cytokinin is transzeatin or isopentenyl adenine. This we have already seen in our previous slide itself. Transzeatin and isopentenyl adenine. Okay. If excess of cytokinin will be produced, how it will be catabolized? This is another most important thing. If excess of cytokinin or cytokinin is produced, that will be targeted by cytokinin oxidase gene. This is very important. Cytokinin oxidase gene. Okay. This what it will do means it will convert cytokinin into inactive form, that is adenine and 3-methylene butanol. So the cytokinin content will be decreased here. Clear? So CK gene is the primary reason for converting the transzeatin to 3-methylene butanol. As I already told in our previous lecture, the genome-edited line, okay, first world genome-edited rice variety, that we already know, CR-DAN 100, okay, and Pusa DST1, okay. In the CR-DAN 100, they have targeted this gene, CKX gene, okay. That's why I am writing here. The CKX gene was targeted in CR-DAN rice variety. This is first world genome-edited rice, okay, genome-edited rice through CRISPR-Cas genome editing technology, okay. If you mutated this gene, so cytokinin will be present in active form, okay. So cytokinin physiological performance will be more, okay. I hope this is clear for everyone, okay. CK signaling, in CK signaling you have to remember only 2 points. I am not going to see all the signaling pathway. What is the receptor? It will have a membrane bound receptor. Receptor means it's a receptor. It will go and bind with the active biomolecule or signaling molecule. Because of that, signaling will happen. So the receptor is a membrane bound receptor for cytokinin. Okay. 3 hormones will have membrane bound receptor. You just remember here itself. Cytokinin, ethylene, brassinosteroid. These 3 will have membrane bound receptors. Okay. Membrane bound receptor. Ethylene will have plasma membrane, cytokinin will have plasma membrane bound receptor. Ethylene will have ER membrane bound receptors. Okay. Brassinosteroid also will have plasma membrane bound receptor. The receptor name for the cytokinin is AHK2, AHK2, 3 and 4. That means Arabidopsis histidine kinase 2, 3 and 4. You have to just remember this one. I hope this is little much difficult for you guys, but these are the key points they are going to ask in exam. These are the deciding factor also. So no one will write if they ask the cytokinin, what is the receptor of cytokinin? If you know that answer, you are going to get 4 marks. So these are the deciding factor for getting top 5 ranks. Okay. So you have to just remember this one. So cytokinin means AHK 2, 3 and 4. Okay. Ethylene receptor or ETR receptor. ETRs are ethylene receptors. Okay, clear. Signaling, I already told, I am not going to discuss about that. That is just for your information. Okay. Now we are going to see the major physiological function. In major physiological function, the first and foremost important thing you have to remember is cytokinin is a cell division hormone. Okay. It's a cell division hormone. Okay. It will delay senescence. The delaying of senescence by cytokinin is known as Rick-Mang-Lang effect. Okay, Rick-Mang-Lang effect. This is another important objective point you have to remember. Delay of senescence by cytokinin is known as Rick-Mang-Lang effect. Okay. Another most important thing, it will be involved in root nodal development, nutrient allocation. It will also contribute for environmental signaling and pathogen responses. These are some of the important key points. Okay. I hope cytokinin is clear for everyone. Now we are going to see about ethylene. Ethylene, the most important thing, it is the only gaseous hormone in plant. Okay. It's a gaseous hormone. Okay. The physiological function we will see in next slide. Okay. So You have to remember it's a gaseous hormone. The biosynthesis of ethylene is very important. The precursor for ethylene biosynthesis is methionine. Okay, methionine is the precursor. Okay, that will lead to formation of S-adenosyl methionine. From here, ACC will form, and the ACC will be converted into ethylene. Here, what the thing you have to remember is Yang cycle will happen. Yang cycle, Y-A-N-G, Yang cycle. Okay. If you see, methionine is a sulfur-containing amino acid. Okay. But this ethylene, if you see the structure of ethylene, CH2 double bond CH2 will be there. Here the sulfur is not there. So, for sulfur recycling, Yang cycle will happen. I hope this is clear. Yang cycle means for recycle of sulfur will be happen. Okay. The key most important thing is different enzymes are there. ACC synthase and ACC oxidase are the enzymes involved in ethylene biosynthesis. Here the question what they will ask, I told 4 important things. What is the precursor for ethylene? That is methionine. Why Yang cycle is happening? That is for recycling of sulfur. And the enzymes involved in ethylene biosynthesis is ACC synthase and ACC oxidase. These are the things you have to remember. Okay. About the signaling, I already told you. Just have to remember the receptor for ethylene is ERS1, ERS2, ETR1, ETR2 and EIN4. These are the examples of receptor signaling. You don't want to read, just you have to read about the receptor. Okay. Now we are coming to the most important thing, the physiological responses. From here, they are repeatedly asking. From ethylene, they are repeatedly asking question. We are going to see now from ethylene, repeatedly they are asking. First thing, fruit ripening will be there. That will be caused by ethylene. Okay. Senescence and oxidation, flower senescence, this you will write. The most important thing, triple response is the specific symptom for ethylene. Triple response is specific physiological response for ethylene. Okay, this is very, very important thing you have to remember. Then another most important thing, advantageous root formation under flooding condition. This is another important thing. Another thing, female flowers will be induced in cooker bees. This is also another example for ethylene physiological function, and parenchyma tissue formation will be there under waterlogging condition. This is also another example of ethylene physiological function. Now we are going to see about abscisic acid. Important point only, it's a stress hormone, also called as dormine, and most important thing, it is a sesquiterpenoid compound, that is 15 carbon compound. Gibberellic acid is a 20 carbon compound, that is diterpene compound. One more compound is the tetraterpene, that also we are going to see. Another compound is there, that is triterpene, that also we are going to see in upcoming slide. Okay. The biological active form of abscisic acid is cis form. Biologically active form of cytokinin is trans form. This is also another important point. They can confuse. Okay, they can confuse you. Okay, which is the biologically active form of abscisic acid? Biologically active form of cytokinin. Apart from that, biologically active form of brassinocerid also they can ask. Brassinocerid also cis form. Okay, brassinocerid also cis form. These are the key points you have to remember. That's why I am repeatedly telling you. Okay, if you see the biosynthesis, sesquiterpene is a 15 carbon compound. It will be formed indirectly by carotenoid cleavage. Okay, you have to remember this. This is the most important thing. Carotenoid cleavage, indirect pathway of carotenoid cleavage will be involved in synthesis of this abscisic acid. Abscisic acid is a 15 carbon compound. The most important thing, another thing what you have to remember is the primary enzyme, that is one enzyme is there that is specific for this abscisic acid biosynthesis, that is NCED. Okay, NCED, 9-cis epoxy carotenoid dioxygenase. This you should never forget in your life. Okay, as a biochemist or physiologist or biotechnologist, you should never forget. So, NCED, 9-cis epoxy carotenoid dioxygenase, is involved in ABA biosynthesis. Okay, very, very important thing. If an excess of ABA will be formed, that can be converted into phasic acid or dihydrophasic acid. This is degraded product. This is also another important thing. Okay. At last, we are going to see tabular form, that will be easy for you. Okay, just you see once. Okay, the physiological roles, if you see the major physiological role only I am going to see. ABA is a stress hormone. It will induce stomatal closing, and most important thing, vivipary. Vivipary means what? The seed will germinate in the plant itself. That means a cob is there. In cob itself, the seed started germinated. Because of what? Low concentration of abscisic acid. So, ABA will prevent vivipary. Okay, vivipary will be prevented by ABA, and most important thing, concentration of ABA and gibberellic acid will determine the germination and dormancy rate of the seeds. If high amount of gibberellic acid is there, means germination will happen. High amount of ABA will be there, means dormancy will happen. This is also another key point you have to remember. Okay, ABA signaling is not important, just you have to remember the receptor, pyrobactin like receptor, RCR and PYL are the example of receptor. This you just see once, okay, this may help you, okay. Now we are going to see a brief discussion about all the phytohormones, whatever we have discussed about the major hormones, okay. Auxin, discover scientist FW Wendt, gibberellic acid Kurosawa, Skoog Miller, cytokinin, Nelsibo, ethylene, Addict for ABA. Okay, other names we have given. This is very important. Okay. Synthesis. Tryptophan is the precursor. Geranyl diphosphate or IPP or acetate or precursor. AMP ATP will be the precursor for cytokinin. Methionine will be precursor for ethylene. Carotenoids will be precursor for ABA. And where the site of occurrence is there, just I have listed here. You see once. Okay. And another most important thing, what are the major physiological functions? Cell elongation, apical dominance, auxin. Stem elongation, seed germination. Gibberellic acid will be there. Cell division and delay of senescence. This I already told. Lichman-Lang effect. Cytokinin. Ethylene means fruit ripening. This is a universal thing. ABA means stress hormone. Then receptors I have just given here. You just see one stick. How it is conjugated and degraded, that also I have given here. Any doubt is there in these 5 hormones? Any doubts? Kindly let me know. Any doubts is there? I hope it is difficult. I know it's difficult for remembering everything, but you have to remember. These are the key point. Okay, okay, no doubts. Vetaraj, no doubts. Okay. Any other? Safura, Udayalakshmi, Rama, Virat. Any doubts? No doubt. Okay, we are going to see now minor plant hormones. After that, we can have one break. Okay, minor plant hormones. This will take just 15 minutes. Okay, what are the minor plant hormones? We have discussed the major plant hormones now. What is ethylene, gibberellic acid, auxin, cytokinin, everything we have discussed. Now we are going to see about the minor plant hormones. Okay, see 5 minor plant hormones are there. Okay, brasinosteroids, salicylic acid, strigolactone, polyamines, and jasmonic acid. These are the minor hormones. In this thing, again I am saying what are things you have to remember. What is the major physiological function? Precursor, hormone signaling, how it is happening, what are the receptors. Then bioassay, if any bioassay is there, means that bioassay. Okay, these are the important points you have to remember. So, brasinosteroid, the name itself is there, it's a steroid hormone. Okay, brasinosteroid is a plant steroid hormone. Okay, first they have isolated from pollen seeds, pollens of Brassica napus. This they asked one time question. Okay, whether they have isolated from Brassica napus or other species they have given here. Brassica nigra or Brassica indica, like that. They have given the species name here. Okay, so Brassica napus, from there only they have first isolated this BR. As I already told, the cis form is the biologically active one. Okay, this we have already discussed. The precursor, you have to just remember the precursor. Precursor for BR synthesis is campestenol. Okay, campestenol is the precursor for brasinosteroid, and the first biologically active BR synthesis is what? Brasinolide. These 2 points only you have to remember in this. Okay, campesterol is the precursor. First biologically active BR formed is brassinolyde. Brassinolyde. Okay. Now we are going to see the major physiological function. In major physiological function, first we are going to see the typical or what are the specific function for this brassinosteroid. The most important thing you have to remember is vascular differentiation. That means particularly xylem formation will be done by brassinosteroid. And another two most important thing is there, cellulose microfibril arrangement. Cellulose microfibril arrangement will be regulated by this brassinosteroid, and cold tolerance will also be given by brassinosteroid. Remaining everything is just for your understanding only. Okay. So the most important thing is what? Vascular differentiation, cell elongation and cell division, cellulose microfibril reorientation and orientation, then cold stress tolerance. These are the major physiological function. Then if you see about the signaling, I already told BR receptor is a plasma membrane bound receptor. You just have to remember the receptor name BRI1. This thing only you have to remember. Remaining, don't confuse. Okay, this is highly sufficient. Okay. Now we are coming to jasmonic acid. Okay. The jasmonic acid was first isolated from jasmine oil, that is Jasminium grandiflorium. First isolated from jasmine oil of Jasminum grandiflorum. Okay. The most important and repeated question they are asking about jasmonic acid is, it's the precursor for jasmonic acid is fatty acid. If you say particularly, it's alpha linolenic acid. Alpha linolenic acid, that is 18 carbon fatty acid with 3 double bonds will be there. This is the example for precursor for jasmonic acid biosynthesis. If you see tryptophan, 2 amino acid will act as a precursor. Tryptophan for oxine, methionine for ethylene. And for fatty acid, if you see, jasmonic acid will be synthesized from this alpha linolenic acid. It is a major stress hormone. This also you have to remember. If you see the synthesis of this jasmonic acid, it will be happened in 3 organelle. First is chloroplast, second is peroxisome, and third is cytoplasm. This you have to just remember. Okay. Just remember, you don't want to go deep into these things. Okay. So biosynthesis we have seen. Okay. What is the major physiological function? I already told it is a major hormone against stress. If you see particularly against insects. Okay. How it is giving resistant against the insect means it will secrete protease inhibitors. Okay. Protease inhibitors will be synthesized. This will be, if that insect will fed on this will act as a feeding deterrence. Okay. So by this way, it is giving insect resistant. Okay. Then another thing is wound response signaling will be done. This is the key important point. Okay. Again, herbivory means the most important hormone is jasmonic acid. Against virus and pathogen means salicylic acid. That we are going to see in the upcoming slide. Okay. Again, virus and pathogens means salicylic acid. Okay. Now we are going to see about signaling. The signaling, the most important thing you have to remember is receptor. Receptor is what? Call1 is the receptor. See, these are the key points you have to remember. Call1 is the receptor. How the mechanism will happen means, like if you see in auxin, OX-IAA will act as an inhibitor. Like here in jasmonic acid, JAS will act as an inhibitor. If this JAS is removed, the transcription factor will go and bind. The JA responsive genes will be active. Okay, I hope this is clear for everyone. This is just a revision. Okay, now we are coming to salicylic acid. Okay, salicylic acid is a phenolic plant hormone. That means it can be synthesized from phenylalanine amino acid. Okay, phenylalanine or tyrosine amino acid. As I already told, this will be specific again pathogen attack, virus or pathogenic attack. If there means salicylic acid accumulation will be there. Okay, so it plays a primary role in plant immunity. This is also another important point you have to remember. Then systematic acquired resistance is primarily caused by salicylic acid. Okay, salicylic acid. I already told the major precursor phenylalanine will be there, or else tyrosine will be there. That will act as a major precursor for this salicylic acid biosynthesis. And if you see the major physiological function, this we already discussed, systematic acquired resistance will be there. Okay, it will enhance disease resistance will be there, hypersensitive response will be there. That means programmed cell death will be there at the particular infection site of this virus or pathogen. Then it can also enhance the synthesis of PR proteins, that is pathogenesis related protein. This is the second topic we are going to see today. Okay. Okay, biotic and diabetic stress we are going to see in the upcoming slides. Okay, this is signaling. Signaling, just you have to remember. See, if salicylic acid is there, means NPR1 protein will go and bind with the target genes. Then the expression of this salicylic acid responsive genes will be enhanced. They can, one time they have asked this in CSIR question, either NPR1 or NPR2, NPR3 or NPR4. So NPR1 is the primary activator. This will act as a transcription factor. It will go and bind with the target gene. It will enhance the PR synthesis genes. Okay, I hope this is clear for everyone. Okay, I hope this is clear for everyone. Ranges, okay, please tell cutoff for this year. How we can predict the cutoff? Okay, so that will be decided based on the difficulty level of question paper. Okay, if the question is very much easy, means all the people will go and keep on attempting for 120 questions. That time that cutoff will come more than 340, 350 also. Okay, if question is difficult, means 70, 80 question, maximum 90 question also people can't able to attend. That time cutoff will reduce. Okay, so this will be decided based on the difficulty level of the question paper. Okay. Next thing, don't see, now you don't bother. Your thing is just you have to give your 100% for your preparation. Don't worry about the result. Do your 100% hard work. Okay. Now we are going to see about polyamines. Okay. The name itself there, polyamines. Okay. They are small aliphatic amines that will have more than one amine groups. Okay. So the example of polyamine, what they will ask in this polyamine means, example of polyamines, putrescine, spermidine and spermidine. This is the major important thing. This polyamine primarily involved in DNA packing. This is another important key point you have to remember. And third most important thing, what are the biosynthetic precursor? If you see the biosynthetic precursor, arginine amino acid ornithine will be act as a precursor. Right? These are the key points you have to remember, nothing else. Okay. The major physiological function, I have already told. Among the major physiological function, you have to remember this will be primarily involved in DNA packaging. Okay, DNA packaging. Okay, okay, clear. Next thing, stygolactan. This is last hormone for today's section, but from here sure you will get one question. Okay, this is most repeated question, most repeated topic they are asking from minor plant hormones. Okay, if you see major plant hormones, they will target mostly about auxin and gibberellin. If you see about minor, they will target about stygolactin and brassinosteroids. Okay, so if you see about stygolactans, it's a carotenoid derived plant hormone. I already told one more hormone is there, that is carotenoid derived plant hormone. Okay, this is the important point I want to say. Okay, it is a 40 carbon compound. Okay, 40 carbon compound will be there. Okay, this is a key point you have to remember. In stygolactan, what they will ask means, what are the major physiological functions of this stygolactan? This is the key question they are asking repeatedly. Okay, now we are going to see how this stygolactan are biosynthesis. The major precursor for stygolactan biosynthesis is trans beta carotene. This is the key point you have to remember. Trans beta carotene. The major physiological function, I will write and give to you. Every year they will ask the same thing. They are repeatedly asking. Okay, these are the major important functions of stygolactan. Okay, first thing you have to observe here, stygolactan will inhibit shoot branching. This is the key point. They will ask whether it will promote or whether it will inhibit. So stygolactan will inhibit the shoot branching. It will regulate the root development, promote mycorrhizal symbiosis, and it will promote parasitic seed germination, and it also plays role in stress adaptation. These are the major physiological functions of stygolactan. Signaling is not that much important, just you have to remember what is the receptor. The receptor for stygolactan is D14. How the signaling will be happen means, if the inhibitor will be degraded means that will lead to activation of strigolactin responsive genes. Inhibitor is D53 is the acting as an inhibitor. With this we are completing the plant hormones. Now just we are going to see the differentiation of this minor plant hormone. If you see the differentiation, brasinosteroid is a steroidal hormone, jasmonic is a lipid hormone, salicylic acid phenolic compound, polyamine, it's an amine compound, strigolactin is a carotenoid derived compound. This is highly sufficient. Okay. Then precursor for brasinosteroid is camphorol, jasmonic acid is alpha linoleic acid, corismate or phenylalanine for salicylic acid, arginine ornithine for polyamine, beta carotene for strigolactin. This is just for your understanding. You just see about this one. Okay. And another most important thing, the major functions, cell elongation and division, brasinosteroids, jasmonic acid, particularly again wounding and defense, salicylic acid, pathogens and SAR response, polyamines, this we already discussed. Inhibiting shoot branching is strigolactons. Okay. Then I have given the unique features and major specific roles of each and every minor plant hormones. Okay. You just go through about this one. Okay. This is highly sufficient for covering the plant hormones. You don't want to go on reading VK Jain or Tyson Ziegert. Okay. This is highly sufficient. Okay. Any doubt, students? Any doubt? Kindly let me know any doubts is there. Kindly let me know any doubts are there. Yes, no doubts, no doubts. We can have a break for 5 minutes. Okay. I will see you all at 5:08. Okay. I will see you all at 5:08. Okay. Take break, have a chai. Okay. We will see you at, yeah, we will see you at 5:08. Okay. Whether I'm audible now, students? Whether I'm audible? Kindly let me know whether I'm audible. Yes, kindly reply whether I am audible. Yes. Yes. So far we have discussed the major and minor plant hormones. Okay. The second portion of today's lecture is plant stress. Okay. From this plant stress, sure we will expect one or two questions. Okay. They will ask conceptual questions from plant stress. Okay. So you have to remember, okay, if a stress is happening, what are the major biochemical physiological responses will happen in plant? Okay. These are the key points you have to remember. Okay. For each and every stress, there will be some typical or critical points will be there. That also I will let you know in today's lecture. These slides are highly sufficient to revise the plant stress portion. Okay. First we will see what is meant by plant stress. Okay. So stress is what? It's an external factor that adversely affects the plant growth, metabolism, productivity or survival of the plant will be affected. If you see the type of stress, biotic stress and abiotic stress will be there. Biotic means that will be primarily caused by living organism. Abiotic means that will be primarily caused by environmental factors. Okay. The general effect of stress, this we are going to see briefly for each and every stress. If you see a general aspect, first and foremost important thing is plant growth will be reduced, photosynthesis rate will also decrease, oxidative damage, that means ROS production will be more, apart from that yield will be reduced and altered metabolism will be there. Okay, these are the general responses of plant to stress. If you see the classification, this example is very important. Okay, what are the examples of biotic stress? Drought, salinity, heat, abiotic stress. Drought, salinity, heat, cold, flooding, heavy metals and nutrition deficiency. These are the typical example for abiotic stress. Okay. In biotic stress, if you see fungi, bacteria, virus, nematode, insect, herbivorous, and weeds. This is another important thing. Weed is an example for biotic stress. Okay. Now we are going to see first about abiotic stress, the major abiotic stress, then what are the physiological, biochemical and molecular responses. I am going to see tell the important point only. Okay. So drought stress. Drought means what is a major abiotic stress. This will be caused primarily due to water deficient condition. If the water is deficient in soil, that will lead to drought stress. The major physiological response, first plant what it will do? It will close the stomata to reduce the transpiration rate. And second most important thing, it will reduce the leaf area and photosynthesis. Root architectural modification will be there. That means the root growth will be enhanced to take the water from the deeper soil. So root to shoot ratio will also be altered in drought stress. These are the key important thing. Then relative water content of the plant will also be reduced. If you see the biochemical responses to drought stress, this is another important point. Under drought stress, plant will accumulate osmotic protectants, or we can also say as osmotic adjustment. These are small organic compounds that are soluble in nature. This is very important point. Osmolytes or osmotic adjustments are small organic molecules that are soluble in nature. The example of major osmolytes are proline, glycine, betaine, sugars and polyols. These are the very important thing you have to remember. These are example of osmoprotectants. Then secondary defense, what it will do? It will produce antioxidant defense mediated enzymes that may be either enzymatic method or non-enzymatic method that will scavenge the ROS molecules that are produced during drought stress. Then altered hormone balance will be there. Then membrane lipid peroxidation will be there. These are the major biochemical function. If you see the molecular function, different transcription factor involved in drought responsive genes will be activated. For example, if you see DRED, drought responsive elements, this will be drought responsive element binding proteins will be enhanced. This is the key point you have to remember. Then if you see a hormonal aspect, ABA biosynthesis will be enhanced. This is the key point you have to remember about drought stress. Nothing else. These are the key points you have to remember about drought stress. Coming to the heat stress, we know heat means what? If there is an exposure to high temperature beyond the optimal range, that will adversely affect the plant growth and development and productivity by distributing the cellular homeostasis known as heat stress. And most important and critical point, under heat stress, heat shock proteins will be accumulated. This is another important point. These HSPs are classified based on molecular weight. This is also another point, important point. Based on molecular weight, they have classified into HSP 100, 90, 70, 60 and small HSPs. These are the important point you have to remember. HSP 100, 90, 70, 60 and small HSPs. These are the different classes of heat shock proteins. This will be specifically synthesized and accumulated during heat stress condition in plant. If you see the, then we are coming to flooding. In flooding means what? If an excess of water is there means that will lead to flooding stress. Flooding we can also say as waterlogging or submerged condition. This is very, very important point. Most important thing is what are the physiological response. If a plant is growing under waterlogged condition or flooding condition, what are the major physiological responses will happen? First thing, there will be a hypoxia induced root injury will be there. We know if flooding is there means oxygen deficient will be there. So root injury will be there. That will be lead to the formation of parenchyma tissues. This is very important. If you see, ethylene is primarily involved in this parenchyma tissue formation. This is also another important point you have to remember. Then advantageous root formation will be there. For example, this is a plant. This is the water level. Root will be formed like prop root, like the structure will be formed above the water level. This also you have to remember. Then hyponasty will be there. Then reduced photosynthetic activity will be there. These are the major physiological response. If you see, this is the diagram you just see here. Parenchyma tissue is formed during the waterlogged condition. Then biochemical response, if you see, because of anaerobic condition, lactic acid accumulation will be more. Apart from that, ROS production also will be there. Then antioxidants accumulation will be there. Apart from that, ethylene accumulation will be highly enhanced during flooding condition. These are the key important biochemical response. If you see the molecular response, I already told you, ethylene is the key important phytohormone that are involved in flooding response. So, ethylene responsive factors will be enhanced. Then another most important thing, this is the most important thing you have to remember. Submerged rice will be there. So, submerged rice. There what is the gene or QTL involved is sub 1A, okay, sub 1A. This will enhance the flooding tolerance. Then SK1 and SK2, that means snorkel 1. I am writing here, okay, snorkel, SNORKEL, okay, snorkel 1 and snorkel 2. This will be the gene involved in deep water rise, okay, deep water rise, okay, that will have a tolerance to flooding due to the presence of this snorkel 1 and snorkel 2 gene. I hope this is clear, okay. Now we are coming to salt stress. Salt means what? An excess or high concentration of salts will be there in the soil. So primary response, what it will do? The water uptake by the plant will be affected. If more salts are present in soil, water potential will be what? It will be more less. If less water potential is there, means the uptaking of the water by the plant will be affected. Okay. Another most important thing, this accumulation of salt can lead to iron toxicity also in plants. This is also very important point you have to remember. The molecular response, this is ARS question, okay, but you have to remember, okay. If you see molecular response during salt stress, salt overlay sensitive pathway will happen. SOS pathway will be there, okay. What it will do means, it's a mechanism for removing or excluding the salts whatever accumulated inside the plant cell, okay. It will activate Na plus H plus antiporter. Because of that, whatever the sodium salts that are present inside the cell will be taken to out. I hope this is clear for everyone, okay. Next thing is metal toxicity. Toxic metals, if accumulated, means that will chelate some of the most important enzymes that will lead to decrease in activity of the particular enzymes. Okay. So the most important thing you have to remember about metal toxicity is, during metal toxicity, phytochelatons and metallothioneins, these are the proteins that will be accumulated that will chelate the heavy metals. Because of this, the original enzymatic activity will be retained. Okay. So under metal toxicity stress, there will be a higher accumulation of phytochelatons and metallothioneins. This is very, very important. Okay. Then last two steps, chilling and freezing. Okay. So chilling means what? Chilling means 0 to 15 degrees Celsius temperature will be there. This is very important. Okay. Freezing means less than 0 degrees Celsius. That is below 0 means freezing. Okay. You shouldn't be confused between chilling and freezing stress. Okay. And most important question repeatedly what they are asking from chilling and freezing stress is, during chilling and freezing stress, in the membranes, there will be a higher accumulation of unsaturated fatty acid. Okay. Unsaturated fatty acid accumulation will be more in membranes during chilling and freezing, but saturated fatty acid accumulation will be more in heat stress. This is the key point they will ask. Okay. This is the key point they will ask. The physiological response, this is very much easy only. Membrane rigidification will be there because of chilling. Photosynthesis activity will be reduced. Water imbalance will be there. Wilting and leaf curling will be there. Okay. These are the major physiological function. Molecular response, this is just for your understanding only. Okay. CBF will be induced. That means C repeat binding factor will be induced. Okay. Then cold responsive genes will be activated. This is just for your understanding only. Okay. Then membrane lipid modification, we have already told, desaturase gene will be enhanced. Because of that, unsaturated fatty acid accumulation will be more during chilling and freezing stress. Okay. Desaturase gene enzyme activity will be there. Because of the double bond will be produced. Because of that, higher accumulation of unsaturated fatty acid will be there. Okay. Last flooding, this both are similar only. I told you the temperature is below 0 degrees Celsius, that is known as freezing stress. Okay. In freezing stress, the symptoms will be almost similar. The most important thing you have to remember is antifreeze proteins will be accumulated. Antifreeze protein. Okay. These are primarily rich in alanine molecule and glycine molecule. This they have asked in 2019 JRF. Okay. Okay. Alanine molecule will be rich. Apart from that, glycine molecules also will be rich. followed by threonine. These are the three amino acids that are predominant in antifreeze protein. Okay, antifreeze proteins will be there. Okay, with this we are completing our abiotic stress. Okay, now we are going to see, or now we are going to discuss about biotic stress. Okay, in biotic stress, again I am saying from biotic stress, this is very minor topic only, like biotic stress, but some of the key important points I will tell. These are very important. Okay, if you see in biotic stress, I already told fungi, bacteria, virus, nematode, insect, herbivorous and weeds. These are the causative agents for causing the biotic stress. Okay. Now coming the question, if human body is infected, antibodies will be produced. Correct. If human body is infected means antibodies will be produced. How the plant immunity will be there? If a pathogen is infecting, how the plant's immunity will be there? This is very important point, not only for your JRF examination, for your other life science examination, this will be highly useful. Okay. For example, there are different classes of immunity will be there in plant. Okay. First and foremost important thing is PTA immunity. Okay. Pathogen triggered immunity. Pathogen triggered immunity. That means what? Each and every bacterium or fungi that will have certain cell wall modifications or cell wall will be there. Okay. If a bacteria or some of the fungi will coming and attach on the surface of the plant, the receptor that are present in the plasma membrane of the plant, it will recognize the microbes. Okay, that will recognize the microbes. So once the microbes will be recognized, that will enhance the synthesis of PTA immunity. Okay, pathogen triggered immunity will be synthesis. Okay, because of that, the immunity level will be enhanced. If you see what is PAMPs and MAPs, okay, PAMPs, MAPs, pathogen associated molecular pattern and microbe associated molecular pattern, that are simple. That are what are the cell wall components that are associated with bacteria and fungi molecules. Okay, so this component will be recognized by the membrane receptors. Because of that, immunity level will be increased. Okay, if the immunity level is increased, to fight against this immunity, the bacteria can secrete some of the effector molecules. Okay, so pathogen will release the effector molecules that will decrease the immunity level to below optimum level. Okay, because of the release of effector molecule by pathogen, this immunity was decreased to below optimum level. If the effector molecules are released by pathogen, the plant to fight against the effector, it will produce what? It will produce A-virulence proteins. It will produce. Okay, A-virulence proteins will be produced. Because of this A-virulence proteins, the effect of immunity will be high comparing to the previous thing. This is known as effector triggered immunity. Because of the effector released by pathogen, the immunity level was increased. You just have to remember these 3 points only. Okay. So pathogen triggered immunity, this will be caused primarily due to PAMPs and MAMs. Then effector triggered susceptibility. That means because of effector, the immunity was reduced. And third most important thing is effector triggered immunity. Because of effector, the immunity was triggered. Like this, a cycle of mechanism will be happen between plant and pathogen. Okay. Because what is the importance of this? If you see, co-evolution will be happen. In plant also some of the different mechanism will be produced to tolerate against these pathogens. These pathogens also will evolve in different ways to escape from this immunity. This is known as zigzag model of immunity. This is very, very, very important point. Okay. These 3 points are very, very important. Here I have just listed the brief explanation of this. If you see the PAMPs and MAMs, for example, bacteria will have flagellin, EFTU will be present in bacteria, fungal cell wall are made up of chitin, lipocytosaccharides will be present in gram-negative bacteria, and beta-glucans will be present in fungi and humaceae. This will be act as a PAMPs or MAMPs for enhancing the pathogen triggered immunity. I hope this is clear for everyone. Then another most important thing, systematic acquired resistance. Okay, systematic acquired resistance. I already told. SA, salicylic acid is primarily involved in systematic acquired resistance. Now we are going to see what is meant by systematic acquired resistance. Okay, if a plant is there, I will show the figure that will be easy for you guys. Okay, if a plant is there, this is a plant. If the pathogen is infected at particular tissue of a plant, here the salicylic acid accumulation will be more. Okay, this salicylic acid accumulation, more accumulation of salicylic acid, what it will do means, it will lead to synthesis of mobile signals that will be translocated away from this infected tissue. It will spread throughout the tissue of the plant. Okay, why this is happening means, if this mobile molecule will transfer and go to other tissues, there it will enhance the salicylic acid accumulation. So the resistance will be more before the pathogen is infecting the other tissue. Okay, so phloem mobile signals will be produced if a particular tissue was affected. Thus will be transported to other tissues of the plant. Okay, that will lead to systematic acquired resistance. Okay, now you see the definition. Systematic acquired resistance is what? Long-lasting, broad-spectrum, systematic defense mechanism activated in plants following the localized pathogen infection, typically by biotrophs or hemibiotrophs pathogen. Okay. What is the reason? SAR prepares, systematic acquired resistance, prepares the uninfected tissues for enhanced resistance. I hope this is clear to everyone. I hope this is clear to everyone. Okay. Now we are going to see what are the long-lasting or long distance signal transmission molecules. If you see here, the example of this is methyl salicylic acid will be there, or azelaic acid will be there, glycerol 3-phosphate and dihydroabitol. These are the typical example for the mobile signaling molecule. This also you have to remember. I hope this is clear for everyone. Okay, systematic acquired resistance is clear for everyone. Now we are coming to another important point, hypersensitivity response. If, see, in this previous slide, if the infection is there means it is producing the mobile signal, it will prepare the other parts of the plant for enhanced resistance. Another thing is there, what means, if an infection is there in particular tissue, there can be programmed cell death around this area that can prevent the further spread of the pathogen to other tissues. This is known as what? Hypersensitivity response. Okay. So the definition, if you see, hypersensitive response is a localized programmed cell death that occurs at the site of pathogen infection. I hope this is clear for everyone. It is the hallmark for effector triggered immunity. This is another key point you have to remember. This is a hallmark for effector triggered immunity. If a pathogen is releasing the effector molecule, at that time plant has to increase the higher level of immunity. That time this hypersensitive response will be activated. Okay, this is just a mechanism they have given. This is not that much of important. I hope there is no doubt for biotic and abiotic stress. Any doubts is there? Kindly let me know. Any doubt is there in biotic and abiotic stress? Any doubts? Kindly let me know. Any doubts are there? No doubts. Safura, okay. Any other students? Sweta, Udayalakshmi, yes. Nothing doubt. Okay. Whether this revision is useful or it's boring, just let me know the correct answer. It's useful or boring? Kindly let me know it's useful or boring. Yeah. Actually, if you go and see the pages, if you go and read in book, it will take minimum 2 to 3 years for covering the only plant hormone portion. Okay. So this revision will be highly useful. So don't waste your time. TK, don't waste your time. Others HRC, no sir, except one person, yeah, useful. Okay. TK. Now we are going to see third portion. TK, third portion of today's lecture, phytochrome. TK, phytochrome. See, in phytochrome and cryptochrome, we are going to see exam point of view questions only. What is the important point for phytochromes and cryptochromes? TK. So phytochrome, TK, phytochrome is a what? It's a proteinous pigment. Proteinous pigments that mediate numerous photomorphogenic response in plant. Okay, it's a pigment molecule, proteinous pigment molecule. It will regulate various photomorphogenic responses in plant. Okay, the discovery of this phytohormone was given by Borthwick and Hendricks. Okay, Borthwick and Hendricks. The question what they will ask from phytohormones, that I will say now itself. Okay, they will ask what are the different types of phytohormones. PR and PFR forms will be there. Okay, PR and PFR form. Why they have given the name PR and PFR means based on the absorption light and which region that light will be absorbed. Red and fair red light region, this phytochrome will absorb. Apart from that, it will also show a minor absorption in blue light regions. Okay, phytochromes will also show a minor absorption at blue light region. Okay, the major role of phytochrome is what? It will act as a biological light switch to sense the quality and duration of light. This is the major role. Okay, if the light quality everything is right, means it will enhance the physiological function. Particular physiological function will be enhanced. Okay, I hope this is clear. Okay, and another most important thing I forgot to add in this slide. Okay, it's a dimer protein. Okay, phytochrome is a dimer protein. This is also another important point. Okay, phytochrome is a dimer protein made up of 250 kilodalton size. Okay, this is also another important point you have to remember. Okay, I already told there are two different forms of phytochromes are there. PR form, that is red absorbing form, it will be stable. Sorry, this will be stable, but it is an inactive form. Okay, then PFR is there, that is fair red light absorbing form. This is physiological active form. You just remember PR, PFR, both are interconvertible. This PFR is biologically active form. Okay. And another most important thing what you have to remember means at which range they are absorbing the light. Okay. So the PR will absorb the red light. The range is 650 to 680. If the red light will be absorbed by PR, it will be converted into PFR form. Okay. If fair red light, that is 710 to 740 nanometer, if it is absorbed, means this PFR will be converted into PR form. I hope this is clear for everyone. This is the concept question they will ask from phytochromes. Okay. Now we are going to see the major physiological response. This crop specific is no need, but what are the major physiological response is very important. Okay. If you see the major physiological response, seed germination, most important thing is deethylation. This is primary role of phytochromes. Primary role of phytochromes. Okay. The inhibition of flowering will be there. Leaf primordia and anthocyanin production will be there. Chlorophyll accumulation also will be there. Growth stimulation and chloroplast rotation. This is also specific role for phytochromes. Okay. So the most important question what they will ask means what are the different forms of phytochromes, and another most important thing what they will ask means what are the major physiological response of the phytochromes. I hope this is clear. Okay, this is simple topic only. Next we are going to see about cryptochromes. Okay, cryptochromes. The most important thing, cryptochromes are blue light absorbing pigments. Okay, it's blue light absorbing pigment. Okay, this is the key point they are going to ask in exam. Okay, so plant response specifically triggered by blue region of the spectrum. Okay, this primarily controlled by a group of pigment. These are known as cryptochromes. Okay, this will be found across different organism, algal, fungi, ferns, and higher plant will be there. Okay, unlike phytochromes, These are insensitive to far red light. This is another important point. Okay. So it's a blue light absorbing pigment. That is the key point or takeaway message from this slide. Okay. And another most important thing, what are the physiological functions or what are the major blue light mediated responses? Here they are asking this question repeatedly. Okay. Stomatal opening is a blue light. Stomatal opening is a blue light mediated response. Okay. This will be primarily regulated by cryptochromes. This is the repeated question they are asking. Okay. Apart from that, phototropism will be there. Okay. Phototropism, that is directional growth towards light. Auxin also will be involved in it. Okay. Then stomatal opening, then inhibition of hypocortical elongation. This is also another important point. Okay. Then intracellular movement will be there. Other responses. This is also very, very important. Okay. Sun tracking plants are known as heliotropism. This is another example. Another important definition. Okay. Heliotropism means sun tracking by leaves. Okay. Phototaxis means what? Movement of whole organism or cells. That is phototaxis. Stimulation of carotenoid and chlorophyll. This is very important, students. I forgot to say. Chlorophyll synthesis also will be enhanced by blue light mediated response. That is, cryptochromes will be involved in chlorophyll synthesis. Chlorophyll synthesis, stomatal opening, phototropism. These are the three important points. In phytochromes, deethylation. That is the key important point you have to remember. Okay. With this, we are completing phytohormones. Okay. Then we have completed phytochromes, cryptochromes. Then we also completed biotic and abiotic stress. Okay. Now we are going to start with respiration. Any doubt in physiology questions? I think we have completed almost physiological like questions. Some of the minor things are there that I will cover in tomorrow lecture. Okay. Some of the minor things are there that I will cover in tomorrow lecture. Any doubt is there so far? Because next we are going to start about respiration. Any doubt is there? Okay, no doubts means we will start respiration. Okay. From respiration, sure they will ask one or two questions. Again, I am saying, from metabolism, as a biochemist, as a biotechnologist or crop physiologist, you have to know about each and every metabolism. Okay, how a molecules are converting, how carbohydrate, proteins, lipids metabolism and nucleic acid metabolism is happening. Okay, you don't see for JRF point of view, my suggestion is no one can remember all the enzyme names, but you have to work smartly. What are the key important points you have to remember? If you know what are the things to be read, you are the smart person to crack this JRF examination. Okay, so I have compiled in such a way what are the important points to be remembered in exam point of view. Okay, so don't miss even a single second of a class. Okay, so today the last topic is respiration. I am going to cover the major thing, glycolysis. Okay, then how pyruvate will be formed, how this pyruvate will be converted into other products, TCA cycle. At last I am going to end with oxidative phosphorylation, how ATP will be produced. Okay. Apart from that, some of the minor topics are there. Pentose phosphate pathway I am going to cover. Gluconeogenesis I am going to cover. These are the simple topic. Don't take stress. Okay. Just I am saying the important things. You just have to focus about that. Okay. Then another thing now I want to say. Tomorrow I am going to cover. About metabolism of DNA, RNAs, proteins, lipids and fatty acid. Tomorrow class is very, 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 very important class. Okay, now itself I am saying tomorrow class is very, very important class. Okay, entire biochemistry, around 50% of biochemistry syllabus we are going to cover tomorrow. Day after tomorrow we are going to see about basic of each and every biomolecule. Proteins, amino acid, everything we are going to see. Okay, now onwards we are going to see biochemistry topics, some of the topics of biochemistry. Okay. Coming to the topic respiration. Okay. So respiration means what? Biologically, respiration is a catabolic process. Yes, it's a catabolic process, enzyme control process in which organic food molecules, mainly carbohydrate, proteins or whatever the biomolecules will be there, that are oxidized to produce the energy, that is ATP, along with CO2 release and water formation. Okay. So the general reactions of aerobic respiration is carbohydrate in the presence of oxygen, it will be converted into, glucose in the presence of oxygen, it will convert into carbohydrate, water and energy. This is exact reverse of photosynthesis. If you see, water molecule, carbon dioxide will be binded with the presence of light energy to form carbohydrate. So, just is a catabolic process. Respiration is a catabolic process for degradation of these biomolecules to produce the energy. Okay. The major substrate will be glucose will be there. Okay. There are some of the feeder pathway also. If apart from glucose, if we take some other disaccharides or trisaccharide, how it will be entered to glycolysis, that we have covered in our lecture series. Okay. For example, we are drinking milk. Milk will have lactose. Lactose is made up of glucose and galactose. Glucose and galactose will be there. This glucose can directly enter to glycolysis. How this galactose will be enter into glycolysis? This is known as feeder pathway that we have already discussed in our lectures. Okay. So feeder pathways will be there that I have also included here. You just see that once. Okay. So the primary substrate will be glucose. So oxygen will be required. Okay. So final electron acceptor also will be oxygen. Okay. The products what it will be formed at last is carbon dioxide, water. Apart from that, ATP will be formed. If you see the complete oxidation of glucose, this is another important point you have to remember. If you see the complete oxidation of glucose, we will get around 686 kilocalorie of energy, or else we can see 2868 kilojoule of energy. Now itself I am saying conversion factor for kilocalories to kilojoule. Just you have to multiply by 4.184. Okay, you just remember 4.184 you have to multiply. Okay. Now coming to the first and foremost universal pathway, that is glycolysis. Okay. The most important thing about glycolysis will happen both in presence and absence of oxygen. This is very, very important point. Okay. It will happen in cytoplasm. The organelle happening where the pathway will happen means cytoplasm. It will occur in both presence and absence of oxygen. Okay, this is specific pathway. Even though it is coming under respiration, it will happen both in presence and absence of oxygen. See, it will happen in cytoplasm. It does not require oxygen. That means it will happen in presence or absence of oxygen. Okay, what is meant by glycolysis? That means what? Sugar lysis is happening here. Okay, glycose means sugar, lysis means splitting. So here a molecule of glucose, okay, a molecule of glucose will be converted into 2 molecules of pyruvate. Okay. Glucose is a 6-carbon compound that will be converted into 2 molecules of 3-carbon compound, that is pyruvate. Okay. If you see here in glycolysis, there are 10 different pathways are there. 10 different enzymatic mediated reactions are there. Among them, 3 will be irreversible, 7 will be reversible. Okay, that we are going to see in our upcoming slide. So, the reversible enzyme mediated pathways are regulatory enzymes. Okay, for example, in glycolysis, hexokinase, phosphofructokinase and pyruvate kinase. These are the key enzymes. You just remember hexokinase, phosphofructokinase, and the third most important thing is pyruvate kinase. These are the key regulatory enzymes in glycolysis, and these 3 are the irreversible step in glycolysis. Okay, now we are going to see. If you see glycolysis, there are 2 different phases will be there. First is preparatory phase and second is known as payoff phase. Okay, in preparatory phase, ATP will be utilized. In payoff phase, ATP will be synthesized. The ultimate role of this respiration is what? Degrading the biomolecules to produce the energy. Okay. So you see here, I forgot to say in this, glycolysis we can also say as Embden-Mayer of Parnas pathway. You just remember EMP pathway. Okay. If you see the enzymatic reaction, total 10 reactions are there. This first 5 step will be act as a preparatory phase. Okay. Glucose binding with ATP, that will lead to formation of glucose 6 phosphate. This glucose 6 phosphate will be converted to fructose 6 phosphate. This will be further utilization of one molecule of ATP. It will convert to fructose 1,6-bisphosphate. This will be converted into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase. This is JRF 2019-2021 question. Okay. Aldolase is the enzyme that is responsible for cleaving fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Then isomerase enzyme will act that will convert the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Okay. So glyceraldehyde 3-phosphate will be formed at the end of the preparatory phase. If you see here, 2 molecules of ATP will be utilized. If you see in preparatory phase, 2 molecules of ATP will be utilized. Another most important thing you have to remember is phosphofructokinase. This is the pacemaker of glycolysis. If this enzyme reaction happens, means glycolysis will be further proceed. That I will show why it is happening in the next slide. Okay. So this we have already discussed. Glucose will be converted into 2 molecules of 3-carbon compound. This we have already discussed. 2 molecules of ATP also will be utilized. Okay. The next phase is what? Payoff phase. Here we are going to synthesize the ATP. Okay. Here you see just for the first, the 6th step is what? Glyceraldehyde 3-phosphate will be converted into 1,3-bisphosphoglycerate by the enzyme G3P dehydrogenase. This will be converted into 3-phosphoglycerate, 2-phosphoglycerate. Okay. This will be converted into phosphoenol pyruvate. Phosphoenol pyruvate will be converted into pyruvate. These are the 10 different pathways. Okay. 10 different enzymatic catalyst reaction. The important things we are going to discuss now. Okay. The inhibitor for this glyceraldehyde 3-phosphate dehydrogenase is arsenic. This is very important point you have to remember. Okay. Arsenic is example. Arsenic is typical example for inhibitor of glyceraldehyde 3-phosphate dehydrogenase. This is very, very important. Enolase. Sodium fluoride is the inhibitor for enolase. They have already given here. Sodium fluoride is the inhibitor for enolase enzyme. Okay. I hope this is clear. So, if you see here in the payoff phase, 2 molecules of ATP. See here, if 1 molecule of glyceraldehyde 3-phosphate is going, means 2 molecule of ATP will be formed. If 2 molecules of glyceraldehyde 3-phosphate means 4 ATP will be produced in payoff phase. If you see the net gain, 2 ATP will be utilized in preparatory phase. So total 2 ATP will be there. Along with that, 2 molecule of NADH will be produced. If this NADH will enter to electron transport chain, it will produce 6 molecule of ATP. Total of 8 ATP will be produced from glycolysis. I hope this is clear. So total of 8 ATP will be produced in glycolysis. This is very important. If the glycolysis is happening under anaerobic condition, in anaerobic condition, the NADH will not be reoxidized. In that case, only 2 molecule of ATP will be produced. I hope this is clear. This is repeated question also. Under anaerobic condition, only 2 molecules of ATP will be produced. Okay. This is summary just we have already seen completed. Okay. We know at the end of this glycolysis, pyruvate will be formed. What is the fate of this pyruvate? If aerobic condition is there, means this pyruvate will be converted into acetyl-CoA. This will be converted into carbon dioxide and water molecule. So much of ATP and NADH and FADH will be produced. That will be entered into electron transport chain for increasing the ATP synthesis. If anaerobic condition is there, means there are 2 different fates are there. Okay. First is ethanol fermentation. That is fermentation method in where the pyruvate will be converted into ethanol. Then another most important thing is lactate fermentation. Here the pyruvate will be converted into lactate. Now we are going to see what are the questions they will ask from this slide. Okay. So the major important thing is enzyme. Okay. How the pyruvate is converting into ethanol. Okay. How the pyruvate is converting into ethanol. How the pyruvate is converting into lactate. So pyruvate is converting into lactate by lactate dehydrogenase enzyme. Okay. Lactate dehydrogenase enzyme. This pyruvate is converting into ethanol by two different enzymes. Two different enzymes will be there. First, pyruvate will be decarboxylated to form acetaldehyde. This will be found by alcohol dehydrogenase. Alcohol dehydrogenase enzyme will come. Here, pyruvate decarboxylase enzyme will come. Okay. These are the two enzymes that will lead to formation of ethanol. I hope this is clear. Okay. Alcohol dehydrogenase and pyruvate decarboxylase. Here, what question they will ask means, what are the cofactor for alcohol dehydrogenase? That is zinc. Zinc is the cofactor for alcohol dehydrogenase enzyme. I hope this is clear for everyone. Okay. Now, the pyruvate if an aerobic condition is there means that will be converted into acetyl CoA. This we are going to see now, how the pyruvate is converting to acetyl CoA. If you see, the pyruvate will be converted into acetyl CoA by pyruvate dehydrogenase complex. Okay, pyruvate dehydrogenase complex. Okay, this pyruvate dehydrogenase complex will have three different enzymes will be there. That's why we are saying that as complex. Okay, what are the enzymes? Pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. Okay, these are the three enzymes in pyruvate dehydrogenase complex. This is JRF 2023 question. They have already asked this question. Okay. Then another most important thing, what are the cofactors? What are the cofactors involved in pyruvate oxidation? Okay. Total 5 cofactors will be there. Okay. CoAs, NAD, TPP, lipoate and FAD. These are the 5 cofactors. Okay. Okay. Among them 4 are vitamins. Except this lipoate, 4 are vitamins. Okay. Vitamin B1 is required, TPP will be there. Okay. B2 will be required, FAD will be there. B3, NAD will be the cofactor. B5 for CoAs. Apart from that, lipoic acid will be there. That is lipoate. Okay. These are the cofactors required for pyruvate dehydrogenase complex. Another most important thing, the conversion of pyruvate to acetyl CoA, the reaction mechanism is known as what? That is oxidative decarboxylation mechanism. It is not a decarboxylation. It's a oxidative decarboxylation process. This is JRF 2020 question. Okay. Oxidative decarboxylation. This is very important point you have to remember. Now, the pyruvate is converted into acetyl CoA. This will enter to TCA cycle. Okay. So, the TCA cycle will also happen under aerobic condition only. Okay. In TCA cycle, what it will happen? The acetyl CoA will be completely converted into carbon dioxide and water molecule. Okay. If you see the, again I am saying important things only we are going to remember. Okay. If you see the first commitment step of TCA cycle, okay, tricarboxylic acid cycle, we can also call as Krebs cycle. Okay, Krebs cycle. Okay, we can also call this as Krebs cycle. Okay, see here, acetyl CoA will bind with oxaloacetate. This is 2 carbon compound. This is 4 carbon compound. So 6 carbon citrate will form. This is the example of tricarboxylic acid. You see here, 3 carboxylic group is there. It is an example for tricarboxylic acid. This will be found by the enzyme citrate synthase. Okay. Apart from that, 8 different enzymes will be act in the TCA cycle. Okay. This is very simple only. You know these things. I am going to say the important thing only. Okay. The most important thing is 3 enzymes are there. Okay. Alpha ketoglutarate dehydrogenase, isocitrate dehydrogenase. In this 2 step, CO2 molecule will be released. Okay. Isocitrate dehydrogenase and alpha keto dehydrogenase, alpha Kg dehydrogenase. In this 2 step, carbon dioxide will be released. And another most important thing, in each cycle of TCA, in each cycle, 3 molecule of NADH. This is important. Okay. See here, 3 molecule of NADH, 1 molecule of FADH, and 1 molecule of ATP will be produced. That ATP will be produced by substrate level phosphorylation. This is also another important thing. Substrate level phosphorylation. The ATP that are produced in glycolysis is also substrate level phosphorylation. I hope this is clear for everyone. Okay. Now, we can say this citric acid cycle as an amphipathic cycle or amphibolic pathway. Why means the intermediates of the TCA cycle can be utilized as a substrate for synthesis of other biomolecules. For example, if you see, succinate can be used for the synthesis of what? Succinate can be utilized for the synthesis of parperin and heme compound. Like this, the substrate will be taken away from the pathway. Okay. So, if these substrates are taken away from the pathway, there may be some hole will be there in the pathway. That means the particular component will not be there. How it is replenishing? Okay, how this is replenishing? That pathway is known as what? That is anaplerotic reactions. Anaplerotic reaction. Anaplerotic reaction means what? Pyruvate, it will bind with this other different reaction that will lead to the synthesis of components in TCA cycle. I hope this is clear for everyone. Okay. Now we are going to see the energy calculation. This we have already know. Okay. In glycolysis, 2 molecules of ATP and 2 molecules of NADH will produce. In pyruvate oxidation, 2 molecules of NADH will produce. And during this TCA cycle, each acetyl-CoA will produce 3 NADH, 1 FADH and 1 ATP. If you do the energy calculation, it will produce 32 molecules of ATP. I hope this is clear for everyone. Okay. Now we are going to see about glyoxylate cycle. Okay. In glyoxylate cycle means what? Glyoxylate cycle is primarily involved for conversion of this acetyl-CoA in sugar molecules. Acetyl-CoA to sugar molecule. Here the key question what they will ask means the marker or the specific enzyme for glyoxylate cycle is isocitrate lyase and malate synthase. This is the question they are going to ask from glyoxylate cycle. Okay. This we already discussed. Okay. This we already discussed relation between glyoxylate and citric acid cycle. How the fatty acid will be converted. How the fatty acid will be converted. Yes, Sandeep Kumar. How the fatty acid is converted to sugar molecules we are going to see here. Okay. Next thing is gluconeogenesis. That we already know. Gluconeogenesis means what? Is the process of synthesizing the glucose from other non-carbohydrate source. That is known as gluconeogenesis. It is just a reversible of glycolysis pathway. Okay. These are very important. This is very important and this is very easy topic also. That's why I am moving very fast. Okay. Next and last topic is oxidative phosphorylation. Okay. In oxidative phosphorylation, what the thing will be there means whatever the NADH and FADH produced, that will be enter into electron transport chain. Because of this, ATP molecules will be produced. First, we are going to see what are the different complex of oxidative phosphorylation. If you see, there are 4 complex are there. NADH dehydrogenase, succinate dehydrogenase, ubiquinone cytochrome C oxidoreductase and cytochrome oxidase. These are the 4 different complexes. Okay. Then this is the complex arrangement. Here what question they will ask means how much number of H plus molecule will be pumped by complex 1, complex 3 and complex 4. A total of 10 H plus will be pumped, 4 from complex 1, 4 from complex 3 and 2 from complex 2. That means 4, 4 and 2 from complex 1, 3 and 4. This is very important point. This they have asked in JRF 2024. Okay, JRF 2024 they have asked this question. Okay. Next most important thing, because of this pumping, how H plus is producing. How H plus is producing. Okay. Because ATP synthase complex will be there. This H plus will be pumped through this ATP synthase complex. Because of this F0 and F1 component will be rotated. Because of this ATP will be produced. There is another thing is there, rotational catalysis theory proposed by Paul Berg and Peter Mitchell. He has proposed how the ATP will be synthesis by rotational catalysis. If you see, the beta subunit is the catalytic active site where the ATP and PA will bind, that will lead to synthesis of ATP by conformation changes. This is very important point you have to remember. Okay. This is the last slide. Okay. We are going to see the what are the major inhibitors of electron transport chain. From here we will get one question. Okay. So complex 1 inhibitor is PAR. Okay. Pyridine, amytal and rotenone. These are complex 1 inhibitor. Okay. Then complex 3 inhibitor is what? Mixothiazole and antimycin A. Complex 4 inhibitor is cyanide molecules and carbon monoxide. These are the key points you have to remember. Okay. These are the key points you have to remember. Apart from that, you have to know what is the inhibitors. Inhibitor that will inhibit the ATP and ADP exchange. The typical example is atracycloside. Okay. With this, we are completing our today's lecture. Okay. With this, we are completing our today's lecture. If you have any doubt, kindly let me know. Okay. If no other doubt means, we will see the remaining things in tomorrow's lecture. Thank you, everyone. Thank you.